Okay, let's, I still don't have, I'm not hooked up here yet. I've been reading, it's a dangerous thing, all weekend on TF2D, the pre-initiation complex. I've been trying to figure out how everything works. It's beyond mortal man. But I have a few more insights that I'd like to share with you. There have been developments since the course was last taught, which was last summer, updating the structures of H and contribution of mediator. I'm not sure that I have put them at the right place, but they probably need to be distributed. These new slides need to be distributed between the PowerPoint that describes H, TF2H, and mediator, because the two come together to make the pre-initiation complex. So I'm going to cover them here. They may eventually be split out, but it'll be the same slide. You'll know what it means. I have a lot of work to do before the exam opens at noon Saturday. I've got to give you some clues about new stuff, what to learn is not reflected in homework. I need to put some student learning outcomes and make sure that you are prepared for the assessment. That's a good teaching philosophy. Some people call it teaching to the test. I say, tell the students what's important and assess them on whether they know the important stuff and the details, but don't make it a mystery what's going to be on the exam. Allow a student to learn it and show you that they know it. So this is TF2H, and it had this diamond ring kind of appearance, and this figure was on the exams in previous years. And I'd ask you to, I'd label different parts. What is this? That would be where the kinase and the cyclin would be, the CDK7 and cyclin H would be there. These would represent the two helicases, but you know, how do helicases fit there? And so we know a lot more about it now. For example, these have been some of the questions that I've posed here and to myself. If TF2H is required for melting the promoter, then how come it has two helicases? What's that all about? Bacterial polymerase didn't have anything that you'd call a helicase. It just has sigma with a great long protein alpha helix that just melts into the DNA to transition to open promoter complex. So you have two helices in TF2H required for transcription, and one of them seems to be required more than the other. And then you've got TF2B, which has the B linker, which is supposed to be the structural equivalent to domain 2.3 of sigma, that is supposed to melt the promoter also. What's going on? Who's actually in charge of melting the promoter? So the structure has illuminated this area of uncertainty. And so I'd like to share this because these are developments in the last 6 months. This is the cutting edge. This is as much as Homo sapien, the tailless ape, knows about transcription in himself or herself. So this image is, as they say, so yesterday. This is what TF2H core looks like. This is what the two helicases look like. Helicases and their friends. Notice it looks more like a horseshoe, with this being the open end. And look at that long thing. That is MAT1. Remember that structural subunit of the CAC, of the kinase module? it had a structure unit called MAT1 and cycling the the kinase and the cycling control. That is binding up here. What is all this? What's happening here? It turns out that this whole region right there, and this is the rainbow color configuration. So it starts at one end, I can't remember, it starts red, goes through kind of yellow, orange into blue. So that allows you to kind of at a glance see what is a single molecule. So you see red and it convolutes all around and ends up blue. This is one molecule. This turns out to be XPD, as in delta or dog. XPD. That's one of the helicases. And the other helicases down here goes from red through orange, the yellow, light blue into dark blue. This is the other helicase. That's XPB, Baker, Bravo. D, which is the same as Rad3, and B, Bravo. And these other proteins appear to be structural connectors so you can make a horseshoe. Now, if you look at it in the EM, the previous slide, you can see where this may have at one time, depending on the angle, have looked like a donut. And this does not include the CAC, the part that's specific to its transcription role. This part participates both in nucleotide excision repair and in transcription. I'm going to put a little link here. And this is a great site. If you've got really no life at all and need a way to spend Friday night, this is how you do it. You go right here and you get this space-filling model here and you put it on spin. Whoa, who needs a TV, right? Look at that. Could you ask for anything better? It's raining outside, nice cup of cocoa, staring at your laptop screen there, watching that thing go round and round. Then you decide to stop it. Whoa, enough with the spin. Whoa, let's spin it this way. Hit the spin button. Life is good again. Can you see DNA in here? There's a little bit of DNA in there. It'll come around in a minute. There it is, the two helices. Nope, that's three. It can't be DNA. Oh, another lost weekend. So anyway, this is marvelous. But this goes back to archaea. Almost. The XPB and the XPD, those two magical helicases, come from archaea. Yet another connection to Mama Archaea. And even more amazing is those two helicases contain embedded within them something even more ancient. A domain that first learned how to manipulate DNA. REC-A. Two domains of REC-A, the ATPase domain and the DNA contact manipulation domain of REC-A are embedded within each of those two helicases. Wow, I guess Darwin was right. I mean, we go way back. Our lineage is ancient indeed. So let's take a look at, oh, my little friend, I must leave you now. Let's get out of this. So that link there is on the slide, and I know you'll use it responsibly. So let's take a look at what else is there. Here you see sort of the big picture of H, D, that's Rad3, and you can't see you see MAT1, that's that long helix, you can't see in that projection where B is. And that, see, this would be the horseshoe here, up top, D. The horseshoe's turned around from how it was. D, and then down here, that's B. And then map 1 joining the 2. But the CAC is kind of flexible. I believe that the CAC is really joined to that long tether here. This is not really anchored here. This is the more confirmed interaction with map 1. But CAC is located at the end of the horseshoe. So what does CAC do again? CAC phosphorylates position 5, serine 5 of the CTD. That starts the ball rolling. That triggers the transition from initiation into elongation in the eukaryotic pre-initiation complex. So a little more information. Let's keep going here. XPD up top, B on the bottom. Here's one more thing. It turns out that the helicase activity, the ability to creep along DNA, is not required for transcription for D. That is just there to make structural connections, to make sure everything is aligned properly. In transcription, in nucleotide excision repair, it's a full player. If you put mutations in the interior here where it actually handles DNA and where it burns ATP, you end up with Cockaine's disease syndrome. That's where you have the small children that look like old men. That bizarre thing. Stream UV sensitivity. We know exactly where the mutations map. They map in those domains of Rec A. By the way, do we have other instances of Rec A being in our genomes? Well, it turns out that animals are sort of the exception. We don't, but plants and lots of stuff, fungi, most other eukaryotes do have Rec A in their nuclear genomes, and they caught it from their mitochondria. Now, we got a lot of the genes from the mitochondria migrated to the nucleus, and then they're just exported or imported into the chloroplast or to the mitochondria, but they reside now in the nucleus. And so you sit and you go, how can the Almighty have invented diseases? What good do they do? If I was a preacher man. So here's what they can do. That's how we got here, so he can work his wonders, as they would say, and then the organ would start up and everybody would start throwing their flower hats in the air. We are the process of an endosymbiotic parasitic relationship. That's how we got, and we still do have our mitochondria. Wow. That's a bummer, isn't it? Anyway, they don't tell you that in that way in biochemistry. So let's continue this. Now this is the really neat stuff here. This diagram, you see these things right here? Lobo 2, Lobo 1. What a cool name, right? These are two little parts of XPB, the one that's involved in transcription. They form an ATP-driven ratchet machine. It grabs DNA, it's called a helicase, and you think of helicase as melting, right? Because it binds one strand and it climbs one base at a time and it pushes the other strand out of the way. It's not really a complicated melting operation. It's mainly mechanical, the fact that it can move along a DNA strand and just displaces the other strand as it bumps into it. This is a helicase because it promotes melting, but it doesn't do it directly. This describes why you can have H acting to melt and B acting to melt, because they do it in entirely different ways. This little ratchet machine is crawling along the outside of the closed DNA duplex, and it's grabbing the phosphodiester backbone at the minor groove, grabbing the backbone as a handle and pulling itself along that way on the outside of the DNA, burning ATP as the ratchet makes that kind of movement. How can that promote melting? Because this is fixed, it's not moving along the DNA, it's pulling the DNA into the interior of the RNA polymerase and causing this big bend as the DNA becomes seated in the channel. That bending promotes melting, which is further encouraged and facilitated by the linker of TF2B. So now we can have both TF2B linker promoting melting and this ratchet helicase crawling along the outside of the DNA, jamming DNA into the interior channel of the polymerase in the pre-initiation complex to cause it to bend against the wall. Fascinating. And you see the horseshoe here of TF2H. And you see that this here would be B, XPB, with its two lobes, lobe 1 in the back and lobe 2 on this side, and they are crawling along the outside. And up here you'd have the RAD3. This is the XPD, not directly involved in melting, but there for structural purposes because of, you will see a little later, it helps align other complexes here. You can see it, RAD3, it's helping align the CAC. So it's positioned properly to phosphorylate the CTD of RNA polymerase. So both participate in transcription, but the helicase activity specifically of B is required for the promoter melting aspect. Let's see what else we can see in this thing. Yeah, you see sort of this open, this is mediator here, kind of cradling down on the bottom. It turns out there really is some space between the mediator and the RNA polymerase itself. In fact, you see this black line, this is that unstructured beginning of the CTD. This is coiled like a rope in this space here between the mediator and the polymerase as it gets ready to be extruded through that little opening there. And as it goes through here, it becomes phosphorylated because that's the CAC. It's sometimes shown as a trimer. Actually, 3 copies of the CAC are sitting there, it seems. Here it looks like there are 2, probably 3 together. Yeah, here you see the linker coming off the polymerase in gray. It can occupy this space, but eventually it'll snake up past here and exit and become phosphorylated. Okay, so that is the expanded role we learned from structure in the last 6 months about H. I thought this was the place to let people know in this course, this is what we discuss and study. So more about mediator has come to light, and this is part of the pre-initiation complex also. So let's see if we can sort of see this pre-initiation. How many proteins in the pre-initiation complex? It's not quite 50, about 46 or so, but almost 50. It's a giant thing. Okay, so here you see XPD. So we're looking for a horseshoe here. It's going to be like this. The XPD is there. Where will, if that's D, where is B? maybe you can't see it in this view. There's D. Yeah, you can't see it in this view. But you see mediator, and look at this tail module. To me, that looks like some monster salamander lifting itself from the mud. That's its mouth. It's like these, you know, you see on your front porch and on your bushes, these things called anoles with the red throats. And when they get into mating season, they walk funny, up on their tiptoes, and then just boom, boom, boom. I don't know, I can't read their minds, but there's something on their mind. But this to me looks like one of those in that kind of pose where he's tiptoeing up to his girlfriend. Okay. She's going, go away. I see that. Yeah, okay. Can you find TBP? That's the question she'll ask him. Can you find TBP? Give me a minute. Look at that. Well, that's obviously A because it's yellow. B is always green, so TBP's in red right there. Here's DNA, and notice that big kink that it takes. Remember, TBP puts an 80-degree bend in the DNA. So that would be A right here. Transcription is this way, but notice as you're like on some Busch Gardens kind of ride. You're going to go down the roller coaster here and then come flying out this way into U. See the DNA? A is right there. You're getting ready to go over the hill. B is on the other side, and it's snaking underneath the DNA into the interior of the polymerase. And XPD is up there, and you can't see B here. Tail model. So mediator is kind of on the bottom here. But the tail has two configurations. It's called the up and down configuration. We'll have to see more about that in a minute. Here is, look at the top view. If you're looking down on it, on the polymerase, H is sort of out front. Why would H be out front? That's where the two ratchet domains are pulling, or making contact, so they can cram the DNA into the active site. That's why they're out projected in the front. You see F is sort of all over the outside. E helps attach H. Mediator is kind of down below the head and so forth. So if you take this and you rotate this view, where's TBP? Well, you just look at where the big bend is, for one thing. That's where the big bend is. TBP is not shown here. But in red, that would be B, snaking underneath the DNA into the interior of the enzyme. Here, if this is B in red, this would be attached to the side of the back. TBP is going to be sitting there. And now this gives you a better view of mediator packed on the bottom of this big dirt ball. And I think this could be one of the last ones. Maybe there's another one. This is just mediator. You remember the shrimp picture, the EM, the shrimp? It was kind of crude, and all my slides were based on the shrimp. Is the shrimp still there? Does he still live? Well, it's kind of shrimpish there, isn't it? Say it is. Look, there's a shrimp there somewhere. If you rotate it correctly, this is the pawl, the polymerase, and this is the shrimp, and that's the tail, sort of. And that's H. We've stuck H on the end of it. This looks like that thing in front of Turlington, doesn't it? Now we know what it is. It was before its time. You can go now and just feel like, oh, H. You go back to Mama Archaea. Melt me. Okay, so, no, I wouldn't say that. Wouldn't say that, especially since this is recorded. Okay, so look at the tail. Wish granted. It has these two positions. Tail down, and you go, it looks like it's up to me. It's like Trump world. Everything's backwards. Tail down, tail up. I don't know. That's how they named it. Don't know who's in charge of naming. Probably doesn't have a job now. But this is where the transactivator GCN4 binds. A lot of them bind to the subunits of the tail domain. We're not quite sure what the up-down is about, but in one of the two configurations, probably tail down, it's closer to upstream activators, more accessible for grabbing. So we're just kind of learning about mediator, how it works. We really don't have the full picture. If you say here again, kind of get oriented, mediator head, middle, they run sort of side by side. This becomes the tail. You turn this around this way and stare at it. That's what it looks like, coming up like that. This is the tail. Wow, that must be the tail down position. No, the tail up position, since it's down. Yeah, that's the tail up position because it's down. And where's K? You know, K in yeast is the same as the CAC. That's where the kinase is. And this is where, on the other side, you have the polymerase is in the middle, and the CTD comes out underneath the middle, that's head, middle head, middle, right below in that groove there, and has to snake through that portion to come out and get phosphorylated right there. When will these slides end? I think this is the last one. This shows the hard to assign CAC region. Here you can see kind of the horseshoe. This is H, this is D, rad3, right there. This would be XPB, XPB. The other one, other helicase, this is the one that grabs the DNA. Here you can see the tail. And here you see the CTD, and this actually is where it's attached to the polymerase, and it's snaking up here, going through that little groove, which is right there, this is a groove formed by mediator. This is mediator, this is mediator. And that goes, and the CAC, or TF2K, is right behind here, and that's where it gets phosphorylated. So now we know a whole lot more than we did about how things work in terms of a structural model. And what you need to know, so I can assess whether you've absorbed any of this, is what are you expected to know for exam 4? And right now it's not clear. And so I'm going to work on that, what's tomorrow, Wednesday? I'll work on that tomorrow to update a student learning objectives where I tell you specifically what you need to know. I may include a figure there on the student learning objectives so you can kind of look at some figures. And I'm probably going to ask you stuff like, can you show me where TBP is? Can you show me where the CAC domain is? Very basic things. I'm not going to ask you to memorize the structure. If you had an arrow pointed to that tail in the two configurations, and could you identify that as mediator tail? But it'll be things that are easy to identify that lets me know that you have a sort of a general understanding of the complexity of the pre-initiation complex. Okay, and we've already covered 5, right, mediator formally, so we don't need to do that. And so we need to get into regulation. for today, and I have not had a chance to mess that one up yet. So it's just as it was loaded. Any questions on the pre-initiation complex? I think this is probably deeper into that subject than you will go even in graduate school. So I'm telling you all I know. I don't have any secrets here. This is all I know about the pre-initiation complex. And you don't care. No, I know all of you do. Okay, regulation. So we spent a lot of time on the basics of transcription and what a complicated and marvelous machine it is. The problems of recognizing a promoter, the fact that there are 3 different kinds of RNA polymerases in eukaryotes, and we haven't talked about the fact that there are 2 more in plants, very specialized. May not be able to get to those. So let's look at back off and see real examples of how a gene is regulated. And this time the focus will be on the transactivators. How are they built? What do they look like? And how does all that work? And one of the model systems we'll look at in more detail is the steroid nuclear receptors, how they work. So we've got some sort of bookkeeping definitions to get out of the way. And this is what you already know. This seems to be review here. Basal transcription factors are these TF2, A, B, D, E, H, so forth, F. And they nucleate near the start point for transcription. Activators determine the frequency or amplitude of transcription. And they, of course, activators bind directly to DNA sequence. They may contain within them domains that have activity to recruit basal transcription factors, to recruit chromatin remodeling factors and so forth. But they may just be largely DNA recognition alone. And the only thing they recruit is another complex which has all that other neat stuff. So if they have the whole package, they're called a transactivator. But if they don't have a DNA binding domain and depend on that to already be present at the promoter, and they just simply are recruited to something that's already found the promoter, they are called a co-activator. If they don't have a DNA binding domain themselves, but they still have HAT and all that stuff to activate, they're called, just by definition, a co-activator. The same applies to a repressor. It could be simply a DNA binding domain. It binds a silencing element in a promoter, but it can't do anything until it recruits a complex that in turn has HDAC, for example. The thing that it recruits would be called a co-repressor. The component that actually sits on the DNA and recognizes the silencing element in the promoter would be called the repressor, even though it's not complete without the co-repressor acting together with it. So how do you build one of these things? And that's the Genome Project. We, in that project, demonstrate the modular nature of transactivators. A DNA binding domain is not comprised of helices and beta sheets and other structural motifs that are scattered throughout the linear map of the peptide. Fortunately, they are all clustered in compact modular form. That means a DNA binding domain can be snipped out very cleanly and it will continue to bind DNA. And you can start mixing and matching parts. You can mix a DNA binding domain for one transactivator with the activation affinity side, the part that attracts either basal transcription factors or chromatin remodelers from a second transactivator and blend them together across kingdoms, across species, any way you want to, just by splicing, just a Saturday afternoon in the lab. So this illustrates the DNA binding domain. There's usually some connecting stuff, and then there are a variety of activation or activating domains which may have enzymatic activity directly, but most often they act as affinity sites to recruit larger complexes that contain chromatin modification activity or recruit other things. So here you see in very crudely depicted form, this is the element, this is the DNA binding domain. Usually, transactivators bind as some form of multimer. Dimers, trimers are two common ways they bind, and we talked about that. Just like in bacteria, they're usually binding an inverted repeat here, and since a helix can only recognize one helical face of the helix at a time, it's kind of like a log. You can't wrap the log around the DNA so it can recognize a very long sequence. It can only be plopped down into the major groove, and you can read 5 bases at a time. That's a really simple site. You can create those just by accident all over the genome. So to make it more complicated, you usually have multiple copies of the DNA binding protein, so that now the site is so complicated that it's very unlikely that it will appear just randomly by chance. Another thing you wouldn't have in a bacteria, you have a nuclear localization domain. It's something that is grabbed hold to, and the protein is yanked off as soon as it's made into the nucleus. It's a whole transport process that moves proteins from the cytoplasm where they are made on ribosomes back into the nucleus. And it's very easy to construct one of these. It's just a cluster of basic amino acids. Sometimes it is in 2 parts called a bipartite NLS. Other times it's just a group. If you have 5 proteins in a row and 3 of them are positive, chances are it can act as a nuclear localization domain. And then you have 1 or more activation motifs or activation domains, sometimes called ADs. So we have a series here of DNA binding domains that there's a drill on the campus site. And you should be able to look at these, because I'm going to draw a question from a question bank that'll have this entire group is fair game. You should be able to look at it and say, aha, that's a zinc finger, or this is a basic loop helix, or a B-zip, for example. First thing you'll notice and bemoan is the fact that eukaryotes have so many other additional ways to bind DNA than in bacteria. This has been highly elaborated. Lots of novel structures have been developed in eukaryotes. The first we'll talk about is called a zinc finger, and this is comprised of a zinc, obviously, that sits in the middle of the DNA binding domain, and it can make 4 contacts with helices and sheets, and it coordinates the position of the surrounding peptide so that it forms a DNA binding domain. And they have several configurations. 2 cysteines, 2 histones, coordinated histidines, coordinated by a single zinc ion. An example is ZIF68. Let's see what it looks like. Another zinc-containing DNA binding domain is called a bimetal thiolate cluster. If you're looking at it, trying to identify, it has 2 zinc ions. That's why they call it bimetal. And it doesn't have any histidines. It's just cysteines. Gal4, this very commonly used in the lab's yeast transactivator. And it's used in the genome engineering project, a DNA binding domain, is a good example of a bimetal thiolate cluster. It's related to zinc fingers, but it has 2 zincs. And the third class are all the steroid receptors. Vitamin D is not a vitamin, right? It's a steroid. There's a vitamin D receptor. It's a steroid hormone. We'll get to that in a minute. But the whole class of steroids, you know, progesterone, androgen, glucocorticoid, all those things have a very common, a shared transactivator structure that has zinc in it. But it has zinc used in two different ways. Zinc is used as a DNA binding domain, and it looks very similar to a zinc finger. And it's also used to form the dimer, a protein-protein interaction domain. That's kind of unusual in a transcription factor. A lot of times they use leucine zippers to form the dimer. But nuclear receptors use a second zinc module, and this is the terminology for the steroid. See, the steroid receptors are the same thing as nuclear receptors. Nuclear receptor. Why is that odd? You've said it so much and heard about it so much, it doesn't strike you as odd. Because if you were in the receptor business, you would know that all receptors are on the outside of the cell waiting to receive a signal of some sort. They're not in the nucleus. This is a nuclear receptor, which makes it strange. That means the signal gets to it goes through the membrane. There's nothing out in the membrane to detect estrogen or glucocorticoid. The answer is no. Steroids are membrane soluble. You don't need to have the receptor anchored out there on the membrane. You can have the transcription factor itself be the receptor and change from an inactive or apoptosis state into an active form when it binds the steroid in the cytoplasm, or sometimes in the nucleus. So that's the origin of the name. It should strike you as odd, but if you don't know that receptors are always in the membrane, it's just another name. So this is the structure of a zinc finger. Here's the zinc, and here are the four interactions: 2 histidines on a helix and 2 cysteines on a anti-parallel beta sheet. 2 anti-parallel strands forming a sheet. So an anti-parallel beta strand and an alpha helix coordinated by zinc would be the definition of a zinc finger. And here's an example of ZIF68, and here you see it in action. You see the helix sitting right in the major groove, but look, there's another one, and another one, and another one. That's not fair, is it? This is really tricky. With a zinc finger, you got this little helix. You can only recognize 4 to 6 bases with it, right? So what did nature do? It said, well, I'm just going to put them all together in a string. And now you bind this sequence, you bind this one, and you bind that one, and you can keep going around the DNA because you're not depending on one helix to wrap around the DNA. You've just split your DNA binding domain up into little chunks that you can link together so you can make a really complicated binding site without having to form a dimer if you don't want to. This, with these multiple zinc binding domains in the same protein, some of them have as many as 13 of these linked in a row. It's called polydactyl, a polydactyl zinc finger, many-fingered. These people, they're sick, right? They work in the lab all day, don't get out much, stare at papers all weekend, got 2 minutes left. Okay, a polydactyl zinc finger is like Hemingway's cats. Here's some more depictions of zinc fingers. You know, zinc fingers used to be the wave of the future, and I jumped aboard. My lab was working with zinc fingers. It turns out that there's so many zinc fingers in nature that you don't have to know the PChem or the X-ray crystallography. You just can make a library of zinc fingers, of known zinc fingers, and predict what they're going to bind. And a whole science developed where you could engineer any protein to bind to any address in the genome. Just synthesize the DNA binding site. You could predict it. And it worked. But now there's something even better. But at one time, this looked like this was going to be the way that genetic engineering could really be accelerated, because you could now, we figured out, although we didn't understand it, we couldn't predict it, but we could just look empirically and see these things bind to these known sequences. We could reconstruct and design DNA binding domains, polydactyl DNA binding domains, and we designed some of these in the lab, and they work. Major groove. Okay, so Gal4, you remember this looks like a zinc finger, but it has two zincs. So if you're on an exam trying to tell the difference and you're squinting looking for the little sphere, which means a metal ion, which the only thing it can be is zinc in DNA binding domains, you're going, how many zincs do I see? Look, there are two. Already you know it's a zinc DNA binding domain, and it has to be the one that starts off bimetal and you forget the rest of it. Bimetal has two zincs. Bimetal thiolate cluster. An example is Gal4. It's all you need to look at. And so here you see a dimer. This is one monomer. Here's the other monomer. And they are joined together, if you look at down the top, by a leucine zipper slowly coiling around itself to form a dimer. And here you see a side view of it. Here again, Gal4, and you can see the two dimers interacting with the DNA. Leucine zipper being the oligomerization domain. Here you see, I think you understand what leucine zippers are, but we talked about it. It's a heptad repeat, hydrophobic, at positions 1, 2, 3, and 4, for a total of 7, a heptad, right? This is approximately 2 turns of a peptide helix, but not quite, because it doesn't, the math doesn't work out. It's not an even number of turns. And that's why this ridge of hydrophobicity that runs along the side is not straight up and down, so the two slowly coil around each other as they interact with the hydrophobic surfaces. The heptad repeat, the 1,4 repeat structure of the leucine zipper. Okay, so the last category, and then we'll call it quits for the day, of zinc-containing DNA binding domains. This is the one that just is very upsetting. This is what controls being upset, being in a bad mood, being aggressive. This is the steroid receptor. It controls our lives, our destiny. Here you see one monomer, here you see the other monomer. What you see is a zinc ion. It has something that looks like a zinc finger interacting with DNA. And up at the top here, this is a protein-protein zinc module interacting module, and this is the other one. 2 zinc modules are designed for protein-protein interactions to form the dimer, and the other zinc module is a DNA binding domain that closely resembles a zinc finger. Glucocorticoid receptor is an example, but all the nuclear receptors have this basic pattern. Next time we'll learn how these are activated by a hormone and a specific example of a pathway about how that is done. And then we will go into the epigenetics portion. Yes?
This PowerPoint is different than the one that's posted on campus?
It is?
Yes.
Why didn't somebody come up here and wrestle me to the ground and put me in a burlap sack and throw me in the river?
Yeah, it's very different.
It's very different? You like this one better or worse?
Slides missing where you skip over a bunch of slides.
This one skips over a bunch of slides?
No, there are just a few slides missing.
This has slides missing?
No, wait. Ours online has slides.
Yours has slides missing.
Like the ones that you were just
Oh, so you want regulation light. I'll take a look at it. Maybe that is due because we're trying to move here and do some things. I have 2 versions of it, and I cut out some slides to make it so your heads wouldn't explode. I'll take a look. I'll treat you fairly, but I got to act quickly. Okay, so I'll see you. Is next Thursday the last lecture? And this comes to an end? The last Thursday torture where you fight sleep for 2 hours, seems like 2 days. Well, see you Thursday.
Saying this Thursday is the last lecture?
Is it this Thursday, isn't it? And we say goodbye? Really? And then you can go be doctors? Really?